I'm very excited to have my friend Karim here. He's the co-founder, CEO of InstaDeep, business he started in 2014, I believe, out of Tunisia. Last year, they sold the company to BioNTech, and he's been scaling it as an independent company since then, applications across lots of different areas. And I think a good story of like true entrepreneurial resilience doing this when it's really not cool against all the odds. And I'm very excited to see all the progress that him and his team have done. So thanks for joining us, Karim.
Thanks, Nathan. Hi, everyone. It's a pleasure to be here. And it's really awesome to see how the RAAIS community has grown over time. So, disclaimer, I don't have beautiful, fancy videos, but I'm going to take you into a deep dive of what is going on in genomics. And it's a very exciting time for AI and biology. And really, like, there's a lot going on, and sort of we're going to deep dive into that. Just before that, just to introduce our company, InstaDeep. So my background is I'm an AI researcher at heart. I've been always passionate about AI, so I decided to co-found a company, like Nathan said, in Africa 10 years ago, and as a bootstrap, and we've grown. And I think what's exciting about us is that we are really focused on developing exciting research. We've done lots of research with most of like the leaders in the field on that front, with universities as well. But we really focus on what I call productized innovation. So really like bridging the gap between research and concrete applications. And so we worked with several great partners, Google, BioNTech, who eventually acquired InstaDeep. And so we joined BioNTech group a year ago. And since then, we've been continuing to develop our capabilities along two major focus areas: biology and AI, which is going to be the topic today, but also industrial optimization and solving operational research, combinatorial optimization problem, which will be for another presentation. But now if we deep dive into genomics, why is this interesting? Well, actually the same technologies that power what Synthese has been doing, which is absolutely awesome in videos, is actually revolutionizing biology. And in particular, when you look at genomics, we sort of have the right configuration to make great progress. So on one side, obviously, capacity in terms of compute is growing very quickly, roughly at an order of magnitude every year in terms of like total compute efficiency improvements. And genomics at the same time, I mean, sequencing a human genome whole genome is almost $100, between $100 and $200. So this is becoming real. And the fundamental interest here, and sort of like, I've always been very curious about like what we can do with advanced AI. And genomics is super exciting because until recently, people were talking a lot about junk DNA. Only roughly 1.5% to 2% of the human genome is actually ultimately translated into proteins. And so the rest was kind of a mystery. And we didn't have the right tools to kind of uncover this mystery until recently. And so with the advent of transformers and attention-based models, we have actually what it takes to be able to go and understand what's going on. And the key breakthrough is obviously unsupervised, self-supervised learning. This notion that I'm going to mask some particular components of a sequence and actually be able to reconstitute that, because the model is seeing lots of patterns and is capable of reconstituting what's missing and sort of like denoising the model. And so that's what we've done. And typically we do it with groups of 6 nucleotides. So one nucleotide can be 4-letter, ATCG, and we're grouping them in tokens of 6. We found that to be a good compromise. And what's interesting is we started this work in 2022 when it was very early days. Like people were just scaling transformers for NLP. And we were among the first in the world to actually deploy the technology for DNA. And we've done it in partnership, notably with NVIDIA, trained on the Cambridge one, at the time the UK's largest supercomputer. And the results were super exciting. And I just want to say a few words about like the kind of data that we looked at. So obviously we looked at human genome, but we also did it multi-species. And the idea was like, can we have scaling effects? Are we capable of seeing like patterns across multiple species, different sizes of models, and so on? And interestingly, actually, you do see like great results very quickly. So, I mean, if I just go a few more slides here to show you what's the spirit, we're going to train unsupervised. So this is equivalent of an NLP on like training on the whole internet. But then we're going to refine on specific tasks which are interesting. So that's the supervised fine-tuning. So the question is like, already if I'm training at scale in a sort of unsupervised or self-supervised manner, am I seeing some patterns? And the answer is yes. So this is literally the model trained without any sort of like specific downstream task. And you can see when you do like a TSNE projection that actually areas in terms of sequences are qualitatively sort of like segregated by the model. So it sees introns, it sees intergenic regions, 5' UR. So these are different sort of like components of a genomic sequence, but the model is actually capturing that information. And that's very cool. If I deep dive even further, what's interesting is actually if you examine the different layers and the attention heads of the model, you see that they become sensitive to several critical aspects. For example, the presence or not of enhancers. Enhancers are what is driving the expression of proteins and pushing certain regions. Same thing with exons, areas that are expressed. So you can see those starting to appear with attention heads. So this is really cool. But then you can go even further and develop a set of downstream tasks and literally like predicting what you're interested in. For example, you have this particular sequence. Are you going to have specific features from this, epigenetic marks, promoter locations, enhancer locations, and so on? And interestingly, until then, people were training mostly on supervised only, like labeled data. And what we've seen is that our approach was competitive. And we were very surprised to see that actually like an independent benchmark assessed results, and we had very competitive and sort of like we're top ranked in terms of performance on 18 different downstream tasks. Now, some of the learnings from this, everything scales with data, but even like if you're looking at like human genome kind of tasks, actually having multi-species data is going to be useful. So more data does help, even though it's data from other species. We trained on a total of 850 species, and we got results that improve the prediction on human genome tasks. So that's an interesting factor. The second one here is that the bigger we scale in terms of size, obviously, the better the model is if you can feed it with data. So in a sense, it's really behaving the way you'd like it to be. And so that it means that those tools are going to be extremely powerful to understand biology, to understand the human genome. And when you think about it in the future, this means that we could potentially link special phenotypes like a particular sort of like health status of patients with some genomic aspects. There is a lot we can uncover in the future because those models actually work very well. So that's kind of like the exciting thing about it. So we didn't stop here. The question was, okay, this is all very cool. This is at the DNA level. But you also have models that are trained at the protein level. And this actually, protein language models actually became sort of like more famous, like faster. And we were like, can you do things with DNA? And DNA is more complicated because if you see over there on the top sequence, really like a particular protein is very often assembled from multiple locations on the gene. So the information there, but it's spread out into different exon regions. And surprisingly, on protein prediction tasks, so if you take the amino acid sequence, that's a protein, and you take the coding sequence that corresponds to it, like that you see in nature for nucleotides, like the particular sort of like codons that are being used, you are actually even improving prediction with your DNA models. And that's kind of like surprising. So you're doing better than protein language models with DNA models on some tasks. So that shows you how much powerful information there is in DNA. And despite the additional complexity, you can sort of like extract that. And if you combine, this is very recent research which you submitted last month, so I'm really showing you a bit like what's going on in terms of flavors. If you combine DNA models with protein language models, take their embeddings, concatenate everything, retrain everything together, you actually have here a performance that is exceeding both. So some aspects are captured by DNA, but you still have other aspects, particularly linked to protein structure, which you could have value from a protein model directly. So in a sense, this is telling you that multimodality is going to be important. DNA is a very strong foundation on which to build on. You can get a lot from it. And if you add multiple modalities, you're going to get to even like more competitive results. And I think this is what we're going to see next in biology and AI, this convergence towards multiple modalities, and some of them a bit surprising, like you're going to see in a second. But that's kind of like the spirit of what we're doing here. And interestingly, like you see, performances are increasing quickly. So this is perhaps my message for people who are passionate about biology and AI, about genomics. This is an extraordinarily fast-moving field. We were super surprised by how much progress we've done at InstaDeep with a relatively small team, a relatively small amount of compute, but that's really this passion to drive things, to go and get results, to go and understand what's going on. It's been super cool. So now I'm going to show you sort of like our latest breakthroughs, so where we are and how we can drive this even further in terms of capability and results. So this is work we recently published. It's available online, SegmentNT. And what's the idea here? We are passionate about AI. We know what's going on in other AI fields. And so one idea was like, why don't we use tools that are coming from vision, such as like segmentation, to go even beyond? Because until now, everything I've shown you is essentially, hey, give me a particular sequence and tell me things about the sequence, but like in general, not precisely. And what we want to do is, like in this image here, it's not only say, is there a truck or is there a car, but where is it? What is going on? Is there a red light? Is there a green light, etc.? It's like, but where are they? So if you think about it on the context of a DNA, like a nucleotide sequence, this means trying to get to specific nucleotide level resolution. That would be really cool, and that would be something quite novel. And that's exactly what we did. We took actually our nucleotide transformer, the one that we've shown you before, which is really like giving you embeddings at the level of the whole sequence. And we added a UNet head on top, and importantly, used some annotation data. And the idea was like, can we now make predictions at the level of each nucleotide rather than at the level of the whole sequence? And actually, like in this particular example, we took like 14 particular sort of like features, regulatory features. And so for every nucleotide position, we're doing all of these. And what's amazing is actually it's working fairly well. So this is how it would look like. Here you have a nucleotide sequence. You have gone through data just at the top there. And you see here the predictions of the model. So the model is outputting literally like 14 probabilities at every nucleotide position, and is doing it in a way which is actually high performance. We were able to actually exceed the state of the art on multiple reference tasks. And you can see here, for example, on enhancer prediction in a tissue-specific way, we have great performance. But what's really exciting also is the speed at which it runs. So SegmentNT here, literally, you can process a 30 kilobase per sequence, so 30,000 nucleotides in 0.1 millisecond. So this is super cool. This means, wow, suddenly like you have the whole genome here, or I mean, a part of the whole genome, like a good part already, talking about like dozens of thousands of base pairs, but get information immediately. And if you think about it from the practitioner's point of view, this is a game changer. Like suddenly you're seeing everything like with a high definition camera of some sort. And so I'm very, very optimistic that this pioneering work we've done is going to be super useful. And we're working with colleagues at BioNTech and at other institutions as well to really push what we can do. Just an example of what we can do with this, you could take literally, I've told you earlier that this is multi-species, right? So you could take species that are not very well known, not very well annotated, and just run the model, which is actually high accuracy, and actually populate an immense corpus of data, which is going to be useful for people specialized in agrigenomics or particular species and the like. So I think this is really like like as a researcher, I'm kind of like excited about what's next. But I wanted to finish this presentation on something that is even more surprising and interesting. This is very recent work we've published. And this idea was like, OK, we have multiple modalities. But could we do something with language? OK? And here's the thing. We know with models like GPT-4 and others, and there are so many of them, like Mistral, Gemini, etc., that predict the next word or the next token works really well, and you can take the whole internet and start to have like great language capabilities. But if you think about biology in particular, GPT-4 might read many articles of biological research or medical research, it still very likely won't understand a particular DNA sequence because it hasn't been trained on nucleotides or it hasn't been trained on amino acids as such. On the other hand, you have models like the one I've shown you, nucleotide transformer, which is essentially a BERT model, understands genomics, but essentially is outputting numbers and doesn't have conversational capabilities. So we were like, you know, why don't we bridge the gap between these two? And this is something that hasn't been done so far. And the key idea is, how can we do this efficiently? Obviously, we're not going to retrain at the internet scale. It's quite expensive, and you have many open source models there. So we actually took Llama 2, and we also have Llama 3 in development at the moment. So we take an open source language model. And the key thing here is we take the nucleotide transformer model, which we've shown you is state-of-the-art on genomics tasks. And the trick is I'm going to take the embeddings of the DNA model and project them in the space of embeddings from the language model. So I'm sort of like putting everything on the same footing and retraining on specific downstream tasks. How do I retrain? I retrain by literally asking the questions in plain English language and having actually a position here, the at, where I am putting the particular nucleotide sequence. So that is that part, the at part, is going to call the nucleotide model, extract the embedding, project it in the space. And so everything happens from a language perspective. It's literally outputting text, like decoder style. And for it, it's like basically information that it can understand because we project it in the space of NLP embeddings. And so the amazing thing is now you can ask questions in natural language, but you have a model that is powerful and has been trained on a very large amount of genomics data. To give you an idea, nucleotide transformer, we trained it on roughly 1 trillion tokens, like those 6 MERS that I've shown. The whole internet is in the range of like 10 to 20 trillion tokens. So this is already quite large scale, what we've done. And so blending both together, you're like, OK, this is very cool. Now I can speak to my model, and my model can answer me in English with information, and both information that is qualitative, is there an enhancer there or a promoter there, or what does an enhancer mean if you don't know? You have all the capabilities of language, and on top of it, you have exact information. And it can actually even output like particular granular numerical data as well. We covered that. So the key question is, this is cool, but does it work? Does it move the needle? Otherwise, it's like fancy stuff that's useless, right? And the beauty of it, and this is really like the gist of it, is it actually works. And if you take the same 18 reference tasks that I've shown you before, where the original model, BERT style, pure DNA, was competitive and was the best, we actually exceeded with the English formulation. And I think that's super cool. Why? Because this really tells you something about the power of language to bring a corpus of knowledge and data into a context. We've done multiple ablation studies, and it's not because we increased the model scale or everything. It's really because we formulated the tasks in English. Prior to that, the nucleotide transformer, as I explained, we were like doing supervised fine-tuning for this task or that task. So effectively, it was a separate model fine-tuned for this or that. Here, it's the same model with this English language formulation. So the tasks are all part of the same unified framework. And you have all the power of context that is coming from the fact that, you know, if you're using Llama 3, it's been trained at the whole internet level. And so it has this understanding. So there's a whole richness of context, which I think is super exciting. And this allows ChatNT to be state-of-the-art and beat the native, already high-performance DNA model. So for me, this is a sense of things to come. And if you take that same model, a bit like a spirit of what I said before, and you do the same thing to RNA tasks or protein tasks, you will see that it's actually quite competitive, sometimes matching ESM without access to protein level embeddings, which, as I shown before, will add even further value. So we're quite excited about those breakthroughs. To our knowledge, this is the first work that blends excellence in language and communication and conversational capability with excellence in actually genomics, which is something that can really be used by experts in the field. So we're really like blending this together. And I do believe that's the sense of history. We're going to see a lot more capabilities and multimodality coming to the market in coming months and years. And really, like, this will revolutionize things. I mean, already performance is quite good. You can see here, for example, confusion matrix. You have like here 3 classification. Is this a donor, acceptor, or site or not? And you can see confusion matrix, the performance is quite good and reflects quite well ground truth. You can have a melting point on protein data, and the model actually will answer you numerically with the exact numbers, and that's actually correct and competitive. So we're going to see a lot more of that. And the key question is like how we build this future of AI and biology together. And at InstaDeep, we're very strong proponents of open source. We actually open sourced nucleotide transformer. We open sourced segment NT, which is this revolutionary like single nucleotide resolution model. And we're actually pleased to see that NT, nucleotide transformer, was the most popular, most downloaded model in the world on Hugging Face in March this year. So really, like, there is something to be said about opening models and having many people like play with the models, feed us back, like sort of like critical feedback about what works, what doesn't, and together building from there. And so that's what I see is really like the exciting opportunity in biology. If I were to see where we are in biology compared to where we are in language, I would say we're roughly right after GPT-3, okay? When GPT-3 came in 2020, people were like, oh, this kind of stuff is working, actually. Like, you put a context and you can get results 0 shot. It's kind of interesting. But then in the next 3-5 years, I mean, you see like all the revolutionary breakthroughs that we had. I do believe it's the same in biology and AI. And right now we're in 2020 or late 2020, and GPT-3 came out and people are playing and seeing this thing kind of works. I do believe we're going to see revolutionary results in the future. And this is definitely what we are excited about, working with our colleagues at BioNTech Group, but also the outer research community, whether it's on the AI side of things, but also on the biology slash genomics side of things. And finally, yeah, we're having fun trying to build tools that are useful to others, trying to push the boundaries. For example, in oncology, BioNTech has a great program on personalized cancer vaccines, taking the RNA technology that was so successful for COVID and deploying it on a personalized level using AI, actually AI developed in part by our team, to really help solve cancer and save many lives. So I do believe the future is very exciting. And if you're interested in these things, don't hesitate to reach out, whether for joint collaboration or more. And the goal is to continue to make progress, continue to have fun, and importantly, continue to be useful and a net positive asset to all of you. So thanks a lot, and happy to take questions.